Hello, everybody. This is the review recording for Exam 3, which we'll all be taking a week from tomorrow, which is the 17th. And we begin. Again, the topics this is anatomy of the heart, blood vessels, respiratory, lymph. All things that have been covered. If you didn't see it coming from today, there were 3 separate recordings I had to do in 3 sections with animal interruptions. And that was the respiratory stuff. So we move on. So first you start with the heart. Well, I mean, that's if you were wise enough to complete the take-home test, you got this. Because basically it's heart and blood vessels. I mean, there's a little bit of the other stuff that's on there, but a little eye, a little ear, nothing major. So think of these things. Think of what's in the interior of the heart that we had discussed previously. Okay, this basically everything that's there. Remember, we looked at a lot of this stuff in a variety of ways when looking at the heart that were there, the distinction with pectinate muscles and papillary muscles and chorda tendinae, all of those protrusions, that trabecula carneae, which is really sort of the mounds and valleys and all of that stuff that, the internal meat that kind of looks like a trabecular pattern in the sense it's bigger than you think and has more surface area. So, well, which valve is which is mitral, which is bicuspid is on the left, and that's an atrioventricular, tricuspid's on the right. The vessels that exit the pulmonary trunk into the right and left pulmonary arteries by way of the right ventricle, the less muscular chamber, and certainly the left ventricle is, decidedly more muscular chamber, and voila, you have the aorta coming out of that, and we'll get to the branches and things like that, but basically the aorta and the incoming vessels, pulmonary veins from the lungs, superior and inferior vena cava. Remember, the 2 atria are smaller. Remember, there used to be a channel that passed through those areas called the foramen ovale. We used because it was an oval hole. And remember, I told you about the crystal tang thing is supposed to seal over. It becomes the fossa ovalis that's there. Be aware of the distinction between arteries and veins. Arteries take blood away from the heart. Veins bring it back to the heart. Veins will have valves. Not all of them do, certainly. And they're capacitance vessels since they hold a greater amount of volume. And I think all of that's significant as well as there. Let's see. I'm trying to go through the heart stuff. And again, know what flows into what. What flows into the right atrium. What flows into the left atrium. Those kinds of things, all of which I spent a fair amount of time. Of course, the locations of those valves and the distinction between those valves and the semilunar valves, the aortic and pulmonic, even though they all have 3 cusps. The only valve that only has 2 cusps is the bicuspid or mitral valve in the left and between the left atrium and the left ventricle, just to be very, very specific. The uniqueness of the pulmonary artery and pulmonary veins as to the kind of blood that they have. That's very important as well. What the general anatomy of the heart that's there, the protective nature. It has a certain amount of fat that acts as a protective capacity. I think that's all part of it. Don't forget the coronary arteries. The coronary arteries, again, basically given off first, directly after the semilunar valves. First vessel, heart feeds itself first. You have a right and a left. The continuation of the left anterior is that anterior interventricular artery that's giant. It's so terribly important. Feeds basically the left, the anterior part of the heart, and a fair amount of the left ventricle, since the left ventricle is larger. And then remember what goes around that. There's a circumflex that comes off the left. The right goes around and basically becomes the posterior ventricular artery that has more about the right side, the posterior aspect, and it also has a marginal artery. So remember where they come from. That's important. All that is very, very significant. I'm trying to see if we have any more heart stuff that I can find. Let's see. Again, I think this covered most of it that I've talked about so far. I'm seeing if there's anything else I might have forgot. Oh, valves themselves. The difference between a prolapse valve, the difference between a stenotic valve, which is a narrowed valve, basically a faulty valve just doesn't close properly or sufficiently. So it would be more or less of a leaky valve, and it undergoes a certain degree of regurgitation. Remember, if it's narrow, we call it stenotic, and remember if it's totally lost the flap, so to speak, and they're going back into the atria, now we call that a thing of prolapse. Don't forget the basic structure. What kind of cells are there or located? Three types in the myocardium. You have the actual muscle cells. You have the conductive cells I touched on at the very end, but I don't think there's anything on what's what in there, and I'm going to make sure of that as I finish this. And obviously, you have to have a certain structural cells that make up the framework for them to attach to. So I think that's all part of it as well. Covered that. Oh, don't forget the abundance of things that are available both in the arteries and in the veins about the heart. They're called anastomoses. Remember, the name you see it a lot is called in another mouth, and basically it's a series of ways that blood can find its way to its area or find its way back from the area, even though there may be a blockage or a defect someplace else. And we actually build those, and we build those by way of typically exercise and rehabilitation. The whole idea of cardio and cardiac fitness, we can encourage the formation of collateral channels. Additional pathways would be built to, if we strategically do that in that fashion. Remember, the entry chambers, the atria are small. They have those larger, you saw them on the cow heart, those big auricles, those ear-like sections that you cut through. They're located on both sides, and basically they're just the entry chambers that are there. Cover that. Oh, don't forget the distinction. Remember, you have coronary arteries and cardiac veins, and the veins flow into the coronary sinus, and where that flows into the right atrium. Okay. And I think we're done. I'm ascertaining now whether or not there is nothing on the conductive system that I am seeing so far, and I'm almost done. And that's correct. There is nothing about the conductive, so you don't have to know. We'll save that for physiology with the SA node and the AV node, and the bundle of His, which is the arterial AV bundle and the branches, etc., etc. So you can save that, and don't have to worry about that. Think of that as context. That'll build our way to electrocardiograms. Next is blood vessels. And I mean, you're just going to have to sit there and study. And again, that's part of where that exercise, basically that glorified homework assignment we're counting as a test grade for a lab, effectively helps you review the anatomy. What comes where? Okay. So look at the flow. Take the aorta, for instance. So as we're going in the aortic arch, after the coronary arteries are given off first, then you have that brachiocephalic trunk that's going to serve the right side. It's going to break up into the common carotid. It's going to break up into the subclavian, both on the right-hand side. And then the subclavian will give off the vertebrals, that backup circulation to effectively becomes a backup to the carotids in general, but specifically to the internal carotid as it goes up to the interior of your brain. Then the left common carotid goes up, then go off the aortic arch, then the left subclavian. We have to have that little brachiocephalic, but it's only on the right. The left subclavian and the vertebral arteries are again given off of that same purpose. And remember, the two vertebrals go up the interior portion of the brain stem. When as they reach the junction of the medulla and the pons, they come to the basilar artery. So it's the vertebral basilar, BASILAR, base of the skull, so to speak, or base of the brain, where it got its naming from, and it forms together with the internal carotids. Remember, the carotids, if it's common, they have internal and external. The internal form the circle of Willis, along with the vertebral basilar and the basilar artery entering into it. The other part, the external carotid, basically services the facial areas and the external portion of the head and scalp and neck. Has a lot of branches. It terminates along the temple above your ear, continuing toward sort of the interior portion or your frontal bone area, and it's called the superficial temporal artery. So that's where that comes from. Now we take Now we have the thoracic aorta as it goes behind the heart, not looking at any of the branches there. Once it penetrates the diaphragm and goes through it, now you have, remember, there's only three openings, one for the vena cava, one for the aorta, and one for the esophagus. And we'll see all of those, particularly because the next topic is digestive, and we'll start looking at that more closely as well. So at that point, we have a variety of things that are given off. Most of them are in pairs, with the exception of the circulation going to the gut, to the alimentary canal. You have first the inferior phrenics. They go to the diaphragm, PHR for phrenic, PHR for diaphragm. Okay. Then another set of paired arteries. You have the suprarenals, which go to the adrenal glands, then the wide renal artery, because it's a high pressure filter, then the gonadal arteries. The termination of the abdominal aorta is called, termination bifurcates. There's a tiny one called the median sacral vein that comes off. It's not a big deal. But the major termination is called common iliacs. The internal iliacs are given off. They serve the pelvic viscera, pelvic organs, and then the external iliac goes under that inguinal ligament and forms the femoral artery. The femoral artery carries itself down. It's sort of anterior and medial, but dives down through that opening called the hiatus, and the adductor magnus muscle becomes the popliteal artery, and then trifurcates. That popliteal artery will trifurcate into the effectively the fibular artery and the anterior and posterior tibial. Posterior tibial will go all the way down to the medial and lateral plantar arteries. I'm impressed with myself, I got all that stuff. The other unique part is what happens right after this phrenic arteries. What's there? And we call that the celiac trunk. That's the hepatic artery, the splenic artery, and the gastric artery. So stomach, spleen, and liver. And then as we go farther down, we have below that, then we have the suprarenals, and before we get to the renals, we give off the superior mesenteric, which supplies most of the small intestine, some of the large, and then past the renal arteries, effectively before we get to the gonadal arteries, we have the inferior mesenteric, which is the larger circulation to the colon. Okay, the large intestine, and obviously some structures in the small intestine and related areas that are there. Those are the big deals that are part of it. So that's really the sum and substance of all of this. You have to look at the anatomy of the arteries. You have elastic arteries because they have a tremendously large amount of elastic. You're going to see that plays an important role when we really look at circulation, look at blood pressure, and look at heart function because of the rebound effect. And that's really only in the pulmonary artery and the pulmonary trunk and arteries, the aorta, common carotid subclavian, these large vessels. Okay, big vessels, because that really enhances the flow and if anything, makes it less rough in the sense of it limits the turbulence that a pulsatile system would have. You'll see that as we go forward. We don't have to know that for now. That's a big deal. That's part of it. The muscular or distributing arteries, those are the next one. Those are typically the named arteries other than the ones I've mentioned. They're basically going into those structures that they're named for, the areas around the femur. If you're in the upper extremity and giving off the subclavian, you have the axillary artery, the axilla, the brachial artery, the arm, radial and ulnar arteries. Basically, don't forget those little arcades or arches as they come, both hands and feet. That's when they supply the muscular structures and the other soft tissue structures and supply everything all around those areas. If it's there, that's its blood supply is coming from branches of those. As the branches come off those, they get smaller. We can't see them. They're arterioles. That's really where blood pressure is controlled. They still have a wide diameter, but they don't have nearly as much. They have enough muscle tissue to really squeeze and limit the flow to certain areas. That's how we redirect blood flow. I commented about that earlier. And then we get to the capillaries, which are really nothing more than simple squamous epithelia sort of creating a tube. Maybe there just was even one single large epithelial or several ones. And so it's like if you took something that came in segments, and you could wrap it around, and they were all pretty much uniform. So it's just epithelial cells, and there's really maybe just some connective tissue outside of it. Remember the distinctions in the capillaries. Mostly they're continuous, meaning they have tiny openings for water and for things dissolved in water. That's how the exchange takes place. The oxygen and carbon dioxide can certainly get through the membranes, but the water soluble solutes and nutrients and the waste products are being exchanged through those little openings. And they're fenestrated when we get particularly to the kidney because it acts as a filter. And in the liver, we call them sign disorder capillaries. They have such big gaps. Remember, the blood coming back from the gastrointestinal tract by way of that unique system called the hepatic portal vein is laden. It's filled with stuff you just ate that needs to be processed, and potentially some bad guys in there that need to be addressed. And the liver is the big player for detoxification. Let's see what else is here. And so, basically, it's almost like a tracing question. So if you from the small and large intestine, basically they flow into what's called the hepatic portal vein. It goes to the liver. In the liver, it's where, like I said, percolates or bubbles through the liver, gets detoxified, processed. What's left of the venous blood then has to go back to the inferior vena cava. It does so by way of the hepatic vein, which goes to the vena cava, and then the inferior vena cava goes back to the right atrium. That's the circulation. That's there. Let's see what else. Again, look at the distinction between arteries and veins. Look at those cross-sectional diagrams. Arteries always have more muscular tissue, and they have certainly more elastic tissue. And they're typically wider in the media, the middle layer, because that's where the muscle is. Where veins are generally might have a larger diameter in the external layer, because there they have a certain amount of larger ones, particularly some elasticity, but some give, but they can hold. Remember, they're capacitance vessels. They have to hold more blood. Okay. So understand all of those things. So you can look at those diagrams and trace how blood leaves the heart, whether it's going to the upper extremity, up to the head, or to the lower extremity into what organs. Just kind of go that, trace it with your finger and say it to yourself out loud. You'll get it. So remember, again, like the carotid arteries, the internal goes right up the circle of Willis. The external ends in the superficial temporal artery. Think cranial and facial. That's a good way, I think, to remember it. Veins typically follow the named arteries, particularly the distributing ones. There's jugular veins and carotid arteries, just the way that there's the cardiac veins associated with coronary arteries. So other than that, the names are almost the same. You always have two or more veins. Remember that vena comitantes thing for every named artery. Very rare not to. And you have a superficial and deep system. The superficial ones we use for vena punctures to start IVs because that tends to make tiny clots. They can't travel because they can't get through all those valves and things like that and make their way back into the deep system, which could lead to go through the heart and get to the lungs. We call that a pulmonary embolus, a blood clot that's broken off and traveled. So, and effectively, they're a spare part. They're non-essential. That's the beauty of them. And the one in the elbow is called the median cubital, the cubital fossa, cubital, is that area, that sort of indentation in the anterior elbow. Median means midline, that's where the vein is. That's where you typically do a venipuncture. But know the distinction between the cephalic and the basilic veins. The cephalic is on the radial aspect, the basilic is on the ulnar aspect. Again, they're part of that superficial system, as are the greater and lesser saphenous, saphenous veins that are found in the lower extremity. And they all end up joining the general circulation. Basically, they make their way back either eventually to the popliteal vein and then the femoral vein in the lower extremity into the vena cava itself. Really, I actually probably more subclavian vein than anyplace else in the upper extremity. Well, I'm not going to ask you that. So remember that flow that are there. I'm still looking. Oh, remember, veins consistently have the largest arteries. So the reason they call them vena cava, they're cavernous. the inferior vena cava and superior vena cava have a huge diameter because they can give, particularly the inferior, which carries the most blood. Okay, of the arteries, the largest one certainly is the aorta, and nothing is really that large. It would surprise you. The aorta, you're talking maybe a thumb. The vena cava a little bit bigger than that. Obviously bigger when it's filled. Surprised you. I've seen one. I've seen one that's lacerated. Vena cava. That's another interesting story. You see. Okay. Come there. I am looking. Making sure that I'm just seeing if there's any part of the delay. I'm looking for any of the blood vessels I haven't commented on just yet, and I think I've got them pretty well covered. So we're getting near the end. Okay, so now, unless I miss my guess, let me just go right back. I don't think there's any more blood vessel questions. So the last group of questions are on those last two topics, which are respiratory. You haven't done, so you have to listen to it. Okay. Names that are important to look up. The most inferior cartilage of the trachea, the carina. It's the one that bifurcates and gives off the bronchi. I've said it 63 times. I love lining tissues. The main one for respiratory is pseudostratified ciliated columnar with goblet cells. That's the big player. Where the pharynx is, nasal behind the nose, oral behind the mouth, or posterior to, laryngeal behind the larynx. Pretty straightforward. And what defines the larynx basically is surrounded by two giant cartilages that protect it. The epiglottis is more of a valve that controls flow. That's the only one that's elastic cartilage. Everything else is hyaline cartilage, and those large cartilage are the cricoid and thyroid cartilages. I'm not doing anything on the vocal cords, not my long suit. The tracheal rings, they're shaped, they're C-shaped, they're horizontal, they open in the back. That's in part the distinction between the right and left primary bronchi. The right is more downward, the left has to cross over the heart. That's very, very significant. How many branches of each? 3 and 2, 3 to the right, 2 to the left, because that defines the lobes of the lung. The progression of how, and this is in the lecture, and it's in my notes as well. It's how the cartilage gradually disappears, finally goes from rings to plates, and finally disappears in just little fragments. And when it makes the transition and there's none left, effectively you come to the end of what is defined as the conductive system, the part that moves air in and out, and you get to the respiratory division, from the conductive division rather. And the respiratory division then relies merely on the principles of simple diffusion, because you're right there where the exchange of oxygen and carbon dioxide takes place. That's the respiratory bronchioles. Remember the muscle that lies behind the trachea and that space that's there? That's important. I just went over the conducting division. Where are their rings? So the rings, when you get to the bronchi, are typical circular and interspace. There's really no muscle there until it starts to branch, and it branches about 23 times. Okay. And the lining tissue, remember, is pleura on the external aspect of the lung, and so on the external aspect of the lung itself is visceral pleura. On the wall itself is parietal pleura. There's a type 1 cells, or the basic alveolar cells. That's really where things move in and out. They share a membrane, and they're simple squamous with simple squamous cells. So they share a basement layer, a basal lamina, with the simple squamous cells on the endothelial lining of the blood vessels. And that facilitates and makes the exchange so rapid. That's part of it. The type 2 cells make surfactant. And there's another cell, there's tons and tons of these little macrophages that are sweeping up debris. That's important as well. Almost there. And then lastly, with lymph, we were there for the lymph. That was the lecture we did on Wednesday, the lymphatic area that was covered. Lymph vessels, it's big stuff. Fatty substances, large proteins. It's stuff that realistically can't fit in a capillary. Think of it that way. That's there. because formed elements like red cells, white cells, platelets, the plasma is liquid that fits into the capillary. You understand what's going on. Know the structures that are part of the lymph system. Okay, the big organs, the appendix, the tonsils, the lymph nodes, the spleen, the thymus, by definition, are all part of that system. The thymus works throughout your lifetime, but it shrinks, interestingly enough, starting at birth, and by the time you're 17, it's pretty well shrunken. It doesn't have to be big, apparently, to function. I think it just implies that it carries on a bigger role, in the gestational phase, and becomes more, and as your immune system becomes competent at its role, it doesn't require as much mass to fulfill its role. The spleen, basically, spleen is, first of all, we could do without it if we had to. Secondly, its primary mission is to get rid of damaged red blood cells, much more so than its lymphatic function. That's the big deal. After that, it serves as an area where it can activate, immunologically, we'll learn about where B cells and T cells, the sort of the power arm of what's called your adaptive immune system, becomes activated. But its main player is really all about getting rid of old red blood cells. The liver can take over if somebody loses a spleen, or there's a spleen that has to be removed for whatever reason that are there. The thymus, remember, is behind your sternum. It's all about what are called T cells or T lymphocytes. That's how they get their name. B cells or B lymphocytes come from the bone marrow and mature in the bone marrow. T cells come from the bone marrow where they're born, but they mature and become, we call, competent, okay, in the thymus, okay. It's an endocrine gland, so it makes hormones. It's in the thoracic cavity. All of that is true. And so there's lots of structures that collect lymph that are there. We have nodes, we have the thoracic duct, the right lymphatic duct. All of those are part of it. The spleen, all of those collect lymph. They're all part of that picture as well. And remember the distinction. Remember, lymph is another fancy name called chyme, Chyme. That's the bigger part. Chyle, Chyle, is what lymph is called. Chyme is the digestive stuff. And that's cisterna chyli. That was usually that's there. But as it expands, and if it's not there, it really effectively all that flows into what's called the thoracic duct. That drains, and that's always the term we use, everything but the right side of the head, the right side of the neck, and the right upper extremity. That's the right lymphatic duct. And remember, all of these structures basically have multiple inputs and limited outputs, so it flows a lot through. It's like a funnel. It takes a while to drain, and it's sort of, again, going through all those little nooks and crannies and being acted upon in case there's invaders, and being basically, and it's addressed multiple times to make sure invaders don't get through into the system. And that is everything. So we have concluded the review session, and I will be sending it to you as soon as they say, as it is processed. Good night.